Hello everyone, I am Ajinkya Patil, PhD student from Department of Biological Sciences, ISO. I will be presenting my work on Artocarpus hirsutus and other Artocarpus species, which is entitled as Jack of all trades, the genome assembly of wild jack and comparative genomics of Artocarpus. The jackfruit is one of the unique fruits of the world. It is the largest fruit beared by a tree. They have low-hanging fruits originating from the stem instead of branch apices, as in most other trees. These fruits are rich with protein, flavonoids, glucose, and other volatile compounds, and hence are cherished by people. Its high-quality timber is also used industrially. The jackfruit is also an industrially important plant. In fact, it was the first species from genus Artocarpus to be described, and hence the plants in the genus are referred to as the breadfruits. The breadfruit, in fact, has a very different fruit and taste than jackfruit. The fruit is more starchy and not so sweet. So it's used as a vegetable rather than the fruit. The jackfruit and breadfruit both are well known all over the world and are part of multiple agroforestry projects due to their economic importance. Whereas there is another hidden gem of Artocarpus genus from the Western Ghats of India, called as the wild jack. This plant is typical forest adapted species with long trees, strong taproot system, competing for resources in this dense forest. It bears smaller but numerous sweet nectary fruits with different color than their popular counterparts. This tree has long been a part of culture of indigenous people with multiple uses in food, timber, medicines, etc. But unfortunately, could not get popularized like other two species and is still an underutilized tree. The jackfruit and vine are both native to Indian subcontinent. In contrast, the breadfruit is thought to be native to New Guinea and other oceanic islands. Around 18 out of 70 species of Artocarpus have been reported from Indian subcontinent. But wild jack is the only endemic species to the Western Ghats of the India. The Western Ghats is one of the richest biodiversity hotspot of the world with around 2200 endemic plant species. But the paucity of genomic resources from these species has impeded the conservation efforts from Western Ghats. In India, many short, fragmented forest areas are still conserved due to the religious importance. These short fragments are called as sacred groves. The sacred groves have proved effective in conserving multiple endemic plant and animal species. The wild jack is ubiquitous part of these sacred groves. The indigenous communities have age-old association with wild jack as a multi-purpose tree species. The high-quality timber is first choice for Keralites in building long-lasting structures. The famous snake boats of Kerala are also made of wild jack timber. Additionally, it has also multiple uses in food and medicine purposes. So to provide genomic resource of the wild jack, we sampled the leaves from this sacred grove, sequenced it, and assembled its genome. Discuss the three Artocarpus species significantly in their tree length and morphology, inflorescence type and size, their fruit texture, contents, and sizes. In this study, we tried to understand the effect of habitat and associated genomic changes contributing to their morphological differences. To understand the effect of habitat on these three species, we first use whole genomic data to reconstruct the demographic histories using coalescent method called PSMC. The x-axis shows the time in thousands of years from recent to old towards the right, whereas y-axis shows their respective effective population sizes. As we can see in the top panel, the demographic trajectories for both Indian species jackfruit and wild jack have concordant patterns of population bottlenecks, whereas breadfruit shows contrasting trends. Even though wild jack and breadfruit are phylogenetically closer, but these trees from different habitats underwent variable bioclimatic events and hence have different trajectories. The species distribution modeling also supports these estimations, which led us to conclude that habitat is more important than phylogeny in demographic estimations. We further addressed the genome size differences in between these three species. The jackfruit has 200 MB larger genome than other two species. What could have been the possible reason for this? So for addressing this question, we annotated genomic repeat sequences and estimated their abundances. We find that more than 150 megabase pair of LTR Copia repeat sequences are unique to jackfruit compared to breadfruit and wild jack. By correlating their genome sizes with repeat content in order Rosalis, we found a strong positive correlation between them, which suggests repeat mediated genome size evolution in this order. Elucidate the accumulation timing of unique LTR Copia sequences of jackfruit, we estimated Kimura to parameter distance and built the repeat landscape, which allows us to infer the accumulation landscape. We identify that jackfruit has accumulated unique LTR Copia sequences after the divergence from breadfruit and wild jack, and that is the reason for its elevated genome size. This repeat accumulation might translate into enough morphological characteristics of jackfruit. We further analyze gene clustering, gene family evolution, and signatures of selection of these three genomes to understand the lineage-specific evolution in these three species. The breadfruit showed gene family expansions in multiple mitochondrial and chloroplast genes. Moreover, it showed unique gene clusters of mitochondrial expression regulator mTOR15 and ABCB27, which provides aluminum tolerance to the plants. Considering the volcanic history of the New Guinea Islands, and the soil must have higher aluminum content and more acidic soils. This warrants higher energy production for plant to maintain homeostasis and tolerate the acidic environment. Also, the genes involved in light signaling might have also played a part in these plants to adapt to different environments with variable daylight timings. In contrast to breadfruit, the jackfruit showed unique gene clusters for genes providing plant immunity and genes involved in flavonoid biosynthesis. Also, multiple gene family expansions and positively selected genes explain its phenotypic differences of its fruit contents. The Western Ghats is in contrast to New Guinea, has highly nutrient-rich soil, which are harbored by large amount of bacterial and fungal species. The plants must either acquire resistance to them or form symbiosis. This explains the immunity genes being uniquely identified in them, whereas the flavonoid genes explain their high flavonoid content in the fruits and other nectary volatile compounds. Jackfruit also shows strong selection pressure on circadian rhythm genes, and this might have played an important role in maintaining its larger distribution. The wild jack is different from these two species. Due to its forest adaptation and large height, it has to depend upon different pollinator and dispersal network. At higher heights, only some primates, birds, and bats could be its effective dispersers. To attract to its dispersers, the fruit color is yellowish orange, which could be due to carotenoid biosynthesis gene PDS, which shows strong selection pressure. The long filamentous male inflorescence suggests wild pollinating mechanism, due to which the plants have acquired unique gene clusters to maintain self-incompatibility to avoid high self-pollination in incidences, which mirrors in lectin gene family expansion. Unique clusters and positive selection. Multiple gene family expansions in flower development and plant biotic and abiotic interactions explains its adaptation to the forest environment, and hence this plant has a pathogen-resistant timber which is treasured by the indigenous people. The wild jack is still understudied, highly underutilized, useful forestry, and due to the fragmented forest cover and its increased local timber market, could make it vulnerable to population decline. One of our goals of this project was to spread awareness about this plant and to conserve this forest. We hope that our minuscule effort will somehow help this plant to survive and thrive. Thank you.